Hello, everyone, and welcome back to chapter 12. And where this chapter, we're going to be talking a lot about genetics. And we're going to talk about a very cool phenomenon that we've been observing over the past years, which is the patterns of inheritance. So one thing that we've always observed is that, our kids all look like us. Our cousins, our aunts, our basically uncles, people that are related to us we look similar to each other. And of course, this is a very cool phenomenon that people never understood. And during actually before the 20th century, there were two main concepts for this. And this was that, heredity occurs within species, that I can inherit stuff from, humans, but I don't inherit it from a horse or a cow or a pig. And two, the traits are transmitted directly from parent to offspring. So I inherit my traits from my mom or my dad. And these are two very actually important concepts that are still true. But during that time, they believed that these traits were actually born through fluid, meaning that they were carried through the fluid, and meaning that they were passed through blood. That's what they were thinking, actually. And this is not true. Because if this was true, we would then be literally, all individuals will in some way look alike, meaning that, me, my dad, my grandfather, my great grandfather would look all alike if this was true, and it's not. And to show this experiments, actually, to disprove that that these traits are actually passed by blood, was the first experiment by Joseph Kohlreuter that disproved this. And what he did in 1760, during those times, they were cultivating a lot of tobacco uh, plants, and he would actually get different types of tobacco plants and cross them with each other, so fertilize them together, and he would create a hybrid. So basically, offsprings from two different tobacco plants. And these hybrids did not look anything at all like their parents. So this is where they first disproved that, hey, you know what? It actually, these traits are not being passed through blood here. Not true. And then when these hybrids were actually allowed to actually, fertilize with each others with different other hybrids, they also didn't resemble at all, you know, the they didn't to the parents or anything. So again, they were not being passed. These traits were not being passed through blood. Similar experiment was also done in 1823, but this was done with peas. And peas are a very interesting uh, plant because peas they can actually do something called self-fertilization, which basically what it means is that they can fertilize themselves. Peas have both female and male reproductive organs, so they don't need a partner to create offsprings. They can basically keep passing their own genetics to themselves on and on and on. So they can pass their own traits, with no interference. And to create a specific, actually, let's say, plant that is always giving you when it self-fertilizes you and it's giving you always the same traits, we call those true breeding. Meaning that, and here an example, if I have a green bean and if I basically that green bean every time it self-fertilizes, it always gives me green beans. That means it's self breeding. So what this guy did is he actually got two different strains, one that is basically green and one that is yellow. And these guys are self-breeding, meaning that if the yellow is fertilizing, it only gives you yellow. And if the green is only self-fertilizing itself, it's only giving green. So you got those two self-breeding strains, and he basically made them cross-fertilize. So you can actually make them, come together and fertilize each other. And he first out that the kids, so the first generation of kids, actually had one color, which was yellow. This was very interesting. It's like, okay, they only look like one of the parents, but not the other. So this mixing is not true, really, like of this blood. So that's then it's like, no, this is not true then, most probably. So interestingly, the first generation kids were then led to do this self-fertilization, and then they were giving actually children, so the second generation of kids were actually green and yellow, which was very interesting. They're like, okay, this is very, very weird. Now this green color is appearing up, where in the first generation it just didn't exist, but now it is. So this was, quite cool experiments. and which actually really kind of pushed a very certain individual like Gregor Mendel to do more experiments. So Gregor Mendel is a very fascinating scientist. This guy actually, grew up. I think it was in Belgium, I think, and he grew up to very, poor or peasant parents, and he was actually going to school in a monastery. And basically, he was very smart to the point that they actually sent him to the University of Vienna. And during those times, it was quite hard to get those, especially specifically when your parents are peasants. That's almost impossible. So he actually worked a lot on himself to do so. Sadly, he went to the University of Vienna, but he failed, so he had to come back to the monastery. But in the monastery, he didn't give up, which is really cool of him. He actually loved science so much that he still kept doing experiments in the monastery there, and he really loved a lot TA Knight's actual experiments. So he wanted to keep using peas, and he decided to use peas for four main reasons. First one is that research showed that you can create hybrids of peas, which other plants cannot. Peas you can actually do you get two different strains and can make hybrids out of them. So you can cross fertilize them to create hybrids. Second is that pea have many varieties available, meaning that some peas can be green, some can be actually yellow, some can be wrinkled, some can be smooth, some have flowers that are actually purple, some have flowers that are white, many different traits that he could actually manipulate. Three, peas are small and they're easy to grow, so it actually doesn't take too much time for to grow them and not a lot of effort. And finally, peas can self-fertilize or cross-fertilize. Again, this is the thing I actually was explaining, but let me explain it to you in this really nice kind of picture. So this is basically your pea. So you have this pod, which is over here, and then these are the peas, and then you have these really nice kind of stick figures that are anthers. These are the male organs. These are the ones that are creating the pollen. And then over here you have actually the carpel. Or sorry, the actually it's called the stigma. So the stigma is basically the female actually reproductive organs. So what you can do is you can actually what in one case basically this whole plant could do is that it could shoot the pollen from the anther straight into the stigma actually, and that would actually that would be self-fertilizing. So it would actually pass its own genetics to this stigma, and this would then be able to reproduce. So this is the self-fertilizing. So, but if you want to actually fertilize it with another plant, what you would do is you would cut the anthers of one plant, get another one. So here, this is a purple flower, and this is a white flower piece. It would actually cut the anthers. It would take a bit of the pollen from the white and transfer it to the purple piece. So that's cross fertilization, actually. So that's the nice thing of actually, peas that they can self fertilize and cross fertilize. So Mendel's experiments, the methods were actually quite simple. So what he first would do is we create a strain that was true breeding, meaning that he would create, for example, green peas that every time they self-fertilized, they only had green, and then he would create another one that would actually had yellow peas, and it would every time it self-fertilized, it would only give you yellow. Hence, that's what it means true breeding. So you would actually get he would produce them, and then he would cross-fertilize them. And the cool thing is that he would do this reciprocal process, meaning that here, for example, we're transferring the pollen from the purple to the white. He would do also that, but also the opposite. He would also then transfer the the pollen here to here, basically. So he would do both kind of ways. So that's what it means to do this reciprocal process. So he would then basically make this kind of fertilization, and then he would get the hybrid offspring. So this is the first generation offsprings, and this first generation offsprings, he would let them basically do this self fertilization to give you this second generation of offsprings. That's what he would basically do, and then he would count. He would count, what peas had a specific trait. So I'm gonna go with this again using an example of green and yellow peas. So you would get the green and yellow peas and you would make them cross fertilize. Once they cross fertilize, you get a hybrid, which is basically which is the first generation. The first generation were then led to do this kind of self breeding to give you the second generation of kids. And then he would count actually how many green and how many yellow peas there were. And the counting was super important because the counting actually gave us a certain model that we use still under today, which we'll be talking a lot about. So he would actually cross two peas, two self-breeding peas that had, basically had, a trait and had two variants of that trait. So there were many of them. So in this case, for example, you would have a trait which is the flower color, the seed texture, the pod color, plant height, position, and pod shape. For example, in the flower color, there was a different variant. For example, in this case, it was purple or white. So he would cross peas that have that had a different variant of one specific trait, just to test something, one at a time. That's what he would do. So then he would, cross them, and this would give you the first generation, which we also call them the filial generation or F1. And what he would observe, very similar back to TA Knight, he would observe that in the F1 generation, they only resembled one parent, only one parent. And this is really interesting for him because this kind of told him that, hey, wait, one trait is dominant and the other trait is recessive. So I'm going to repeat this again using an example because I guess examples are really cool. So if you would first start, let's imagine that we're always starting with um, basically purple and white flowers. Okay, so here he was testing the whole trait of basically the flower color, and you have two variants, which was either purple or white. He crossed them. He would then cross them, and then he saw that the first generation, so the F1 generation, were all actually purple, but the white was gone. He didn't see any white. And then he said that, okay, the white is recessive. That trait is recessive, while actually the purple is dominant. So if the purple is present, it's just gonna be, dominant over the white color. There was no intermediate between the two. But when you got basically this F1 generation and you allowed them to self-fertilize, you would get this F2 generation. We call them the second filial generation or F2. But here, interestingly, the white reappeared. And it came out to be this certain ratio, this 3 to 1. So out of four basically the peas that he got, three of them were purple and one was white. So suddenly in the second generation, you would see again this recessive trait, which is the white, appear. It disappeared in the F1, but it reappeared in F2, which is really cool for him. However, so what he saw was that, of course, three-fourths of the plants were dominant forms, so in our case would be purple, and one-fourth were actually recessive. So it gives you this actual ratio, 3 to 1. But then he later discovered that no, this is not true. This 3-1 is not true. It's actually 1, 2, and 1. What he discovered is that one of the plants was a true breeding dominant plant, meaning that this purple plant, which is the dominant plant, was true breeding, meaning that if you took that plant and you kept self-fertilizing it, it would always give you purple. Two of the dominant plants, so two of the purple plants, were not true breeding, meaning that if these guys fertilized, they would give both purple and white. So they were not actually true breeding. They were not true breeding. They were dominant plants, but not true breeding. And then one true breeding recessive plant. So it's a white plant that if you kept actually doing this self-fertilization, it would only give you white. So this is actually a really nice color, a nice figure. So this is what you had at F0, so the parent generation. So you have a true breeding purple plant and a true breeding white plant. So this guy, the purple plant or the purple peas, this guy, if it self fertilizes it, only creates purple, and if this self fertilizes it, it only gives white. Hence, this is what it means true breeding. So if you cross fertilize them, the first generation, as we said, were all looking like the purple. So the white disappeared. But then when these guys were able to self cross, you would have actually, three purple and one white. So there's 3 to 1. But this was not true because it was actually 1 to 2 to 1 because only one of them were true breeding, meaning that every time they self-crossed, you would have purely only um, what actually purple. Well, this purple dominant, for example, this one and this one, once if you do the self-cross, you would give purple and also white. Then you would also have this true breeding white recessive. They would only give you white. So this is what it means by this 1 to 2 and 1 ratio. So the F2 generation, it was disguised in a 1 to 2 to 1 to ratio. So his conclusions here was that the plant did not show any intermediate traits, meaning that each trait is intact and discrete. So, meaning that the purple and the white didn't mix together. No, they came as one entity, and they did not mix and are passed as one entity. This is why specifically in F1 generation, when you they were actually allowed to self cross, white was there because the white genes were there, but they were basically being, you know, the were more dominant than them. So for each pair, one trait was dominant and the other one was recessive. So the purple in this case was dominant while the white was recessive. So when you have basically a purple gene and a white gene, the purple would actually shadow or would actually block the white and be dominant over it. So pairs of alternative traits examined were segregated among progeny and of a particular cross. So meaning that these traits were being separated and given to the kids. And of course, alternative traits were expressed in F2 in a ratio to 3/4 dominant to N 1/4 recessive. That's were his conclusions. And this gave him to give this 5-element model, which we still do use, is that parents transmit discrete factors. So these are genes. They actually give genes that encode for a specific trait. So that trait could be the eye color, that could be the color of the flower, that could be whatever. Those were these discrete factors were the genetics. Each individual receives one copy of a gene from each parent. So we receive one. So basically, we have two genes, and we receive one from our mother and one from our father. Three, not all copies of the genes are identical. So a gene actually represents a trait. So let's see the color. Allele is a different form of a gene, so it's basically the alternative form. So the gene, for example, is the color of the eye. Let's imagine color of the, or let's stick to the flowers. Is the color of the flower. That's the gene. The allele is then basically what color is it? If it's purple or yellow, so it's a different form of it. It could be the white allele or it could be the purple allele. That's what it means. If both alleles are the same, they're basically homozygous. So if they're all both purple, they're homozygous. Homozygous. If they're basically different, so you have one purple and one white, so different, they're heterozygous. Alleles remain discrete, they do not blend. And five, the presence of alleles does not guarantee expression, meaning that just because you have a white allele doesn't mean it's going to be expressed, because there is a dominant allele and there's a recessive allele. So this is what he means it. So dominant allele is expressed and the recessive allele can be hidden. And this came out to give us two very main, actually, terms: genotype and phenotype. Genotype is the total set of alleles of an individual contains. So that is you're looking at all of the alleles. Phenotype is the physical appearance. Because as we said, it doesn't mean that if you have this allele like the white color, that you're gonna have white. No. So in the genotype, we might see the white, but in the phenotype we see purple. So it's just purely physical appearance. So really be careful with genotype and phenotype. Genotype is all the sets of alleles. Phenotype is just what is then basically being expressed, and we can see the physical appearance. So now came a very interesting thing, which is the principle of segregation. So as we said, we get genes from our parents, right? So we get them, genes are come in two pairs, so there are two alleles, and we get them from our parents, one from each. And this is coming from the principle of segregation. Two alleles from a gene segregate during gamete formation and are rejoined at random, one from each parent during fertilization, meaning that what happens in meiosis, specifically meiosis 2, is that the alleles are completely separated. Alleles are separated, and they will only join another allele when they're actually, when fertilization happens. That's only when fertilization happens. So this is what basically the principle of segregation means, is that we get two alleles from our parents, one from each, and what happens is that in your, for example, your mother, during meiosis, specifically the egg cells, they go through meiosis and basically their alleles are separated. Then it's the principle of segregation. They're separated, and this now separated or segregated allele will join its other pair of allele only when the sperm cell comes and fertilizes the egg. So there then hence the other copy of the allele or the other allele is coming from your dad. This is basically what it means, the principle of segregation. So one cool thing actually to help Mendel actually really understand a lot, or kind of predict what's going to happen when he crosses hybrids was to do these Punnett squares. And what he would do actually a lot, he would actually, for example, in the case here, he would mark a dominant allele as in capital and the recessive one in lowercase. So here, for example, this is basically the male and this is the female. The male had this actually, for the color, it would have this type of alleles, and the female had this one. So here it actually have this purple and this basically white, which is shown as a lower capital because it's recessive. And what he would do is he would put them basically across in the like kind of x and y axis, and he would do the following. So he would basically, this P would go down here along with this P. And same thing, this P would go down here to here. And of course, here you would actually get, this 3 to 1, sorry, 1 to 2 to 1 ratios. So you have one full PP. So this is actually true reading. You would have basically two P, lower capital P and lowercase p, and then one PP. There's recessive one. So these are what the Punnett squares that actually we still use today.